Howdy, Team Aztecs. Welcome to the final video for Chapter 6, Microbial Nutrition and Growth. So this is going to be a video on counting microbes. So this thing right here is a spectrophotometer. And what you can do is you can get a culture tube. So you get a culture tube like this, and it'll have your microbe in it in solution. So your microbes here. And then you take and you open this lid and you put the culture tube inside, and then you press some buttons here and you get an indirect count in quotation marks of the number of microbes you have in this sample based on the turbidity or the cloudiness of the sample. So you don't always have to count your microbes one by one to estimate the population size or density. You just look at the turbidity. So the higher turbidity, the greater the population of microbes. So what you have in here is you have a light that shines through your sample, and the larger the population of microbes in that culture tube, the less light will go through the population of microbes and come out on the other side, and you can indirectly measure the number of microbes that way. You could also, an indirect way to count your microbes is metabolic activity, and that's the rate at which nutrients are used up by your microbe of interest living in your culture tube, or by the rate at which wastes are produced, or the pH change, or oxygen consumption. All those are indirect ways of letting you know how many microbes you have in your sample. You could also do the dry weight, but that means you'd have to kill the organisms and let them dry out and then measure the weight. Okay, this is kind of a cool way to count your microbes. This is not an indirect method, this is a direct method. So again, here you've got your culture tubes, and then you have your plates with agar, could be nutrient agar in there for the microbes to grow on. But what you do is you fill, how many tubes do we have? 1, 2, 3, 4, 5. So you have 5 tubes, and you fill each one of those tubes with your broth. So this brown stuff is the broth. That's what your microbe of interest will need to survive. And then you take 1 ml of the original culture. So you've got your original culture tube up here that has not been diluted, and you vortex it. You got to vortex it or mix it up, and then you'll use a pipette. You'll take your pipette, 1 ml from your original culture tube, and then you'll put that 1 ml into 9 mils of broth. And we've talked about this before, but that's a 1 to 10 dilution. So 1 ml in 9 mils, 1 plus 9 equals 10. It's a 1 to 10 dilution. And then you vortex this tube. Right here, you vortex it, so you mix up the microbes so they're not just sitting at the bottom. And then you take 1 ml out of that and put it in your next tube, and then you vortex it. That's the 1 to 100 dilution. Anyway, you just keep taking 1 ml out and mixing it in 9 mils. 1 ml out, mix it in 9 mils. 1 ml out, mix it in 9 mils. Until you get, in this cartoon, they went to a 1 to 100,000 dilution. And then from those 5 culture tubes, you take out 1/10 of a mil, that's 100 microliters, out of this sample. And then you use, see this thing over here? Let's look at the hand. So here's the hand. You've got this thing, it's called a cell spreader. So you put your drop, your 1/10 of a mil, aka 100 microliters here, and then you use this guy over here, a self spreader, and you just spread your 1/10 of a mil of broth with microbe in it. And then you do that for each one of your plates. So the 1 to 100, 1 to 1,000, 1 to 10,000, 1 to 100,000 dilution. You spread it out. And then you incubate it for a couple days or however long you have to incubate it for that microbe. And look what happens. At 1 to 10 dilution, you get the colonies, which are, remember, colonies are just giant mounds of, could be 100,000, could be 100 million microbes per colony. And in your first plate, your 1 to 10 dilution, it's too numerous to count. The colonies are too numerous to count. And in your 1 to 100 dilution, the colonies are too numerous to count. And then in this cartoon, this dilution at 1 to 1,000 dilution, it's perfect. It's 65 colonies. That's a high enough number to be representative of how many microbes you have. This 6 colonies, that's probably too low. Yeah, it takes less time to count, but it probably isn't it's not as accurate. And then at 1 to 100 dilution, that's just so diluted you don't get any colonies. So this is a way we actually do in the lab when we're in the classroom. We do this method. Okay. So how do we measure huge numbers of cells, like greater than 2 times 10 to the 4th per mil? You can use direct or indirect method. This method I just showed you is a direct method. It's called the viable plate count. So viable just means the microbes are alive in their colonies, and plate refers to this plate. So you do a dilution series, we just talked about that, to dilute the number of bacteria per mil. Okay, we just went through that. Then you spread 100 microliters, or 1/10 of a mil, onto the culture on Petri plates. I just call them plates, but specifically they're Petri plates. And then you incubate them and you count the colonies. And now here's the formula. You take the to find the original number of bacteria per mil. That's this up here. That's the original sample. You want to know what's the number of bacteria in this original sample, and you want to do it per mil. You take the number of colonies. So in this one, you take 65 colonies times the dilution factor, and the dilution factor is 1 to 1,000. 1 to 1,000 dilution. And then you get the answer. You get the number of microbes per mil of your original sample, which is what you want. In this viable plate count, you only count the plates with between 25 and 250 colonies. So that's perfect. This guy's 65 colonies. If it has fewer than 25, like this one, 6 colonies, then there's a chance that you're going to underestimate the population. The accuracy, how accurate it is to get the number of microbes in your original solution up here, depends on homogeneity of the dilutions. That means when you put the microbe, you take a pipette and you put the microbes in your 9 mils, make sure you mix it really well so it's homogeneous. The ability of the bacteria to grow on the medium, so hopefully this media is what your bacteria likes to live on. The number of cell deaths also feeds into the accuracy of the viable plate count, and also the growth phase of the population. And then, so we talked about this, you need to mix the dilution thoroughly. You need to inoculate multiple plates per dilution. So in this cartoon, it only shows you inoculating 1 plate per dilution, but in theory, you could have like maybe 5 plates per dilution. And you want to use log phase cultures to minimize error. So you want to know that your cultures are in the logarithmic phase of growth. We talked about that last time. More direct methods. So I use this a lot when I worked in cancer biology. So you got your solution of cells here, and you don't know how many are in there. You take a pipette, so this is your pipette, and you put a certain volume of your original sample on this, well, actually, this grid, and under a cover slip. So here's the cover slip up here. It's real thin, and then there's this little port underneath the grid, and you pipette your bacterial suspension in there, and then what you're going to do is each one of these little squares, you're going to count the number of bacteria that you see, and you'll do some fun math, and you can tell how many bacteria are in your original sample. So they call this a Petroff-Hauser counting chamber, and you use a glass slide, we talked about that, with an etched grid. So this is the whole thing is a glass slide. This is a grid. It's etched. That means that these lines are etched or cut into the glass, and then the cover slip goes on top. And then it shows the number of bacteria counted per box. So let's say in this box you count 5 microbes. In this box you count 7 microbes. But you take the number of bacteria counted per box times 1,250,000, and that will give you the number of bacteria per mil here in your original sample. This is a little problematic because it counts both the living and dead cells, and maybe you just want to know the live cells, or maybe you just want to know the dead cells. But if you don't care if they're living or dead, you just want to know the number of cells, you can use this method. Electronic methods to count directly. This is pretty cool. This Coulter counter, we had one of these over in the cancer center, and we'd use a Coulter counter, but they are kind of expensive, and there's a whole bunch of working parts to it. But basically, well, here's the readout on your computer screen. But basically, through a bunch of cool electronics, the Coulter counter counts the cells as they interrupt an electrical current in a small tube that's held in front of a detector. And then it gives you the printout here on the screen of the number of cells that you have. The flow cytometry uses a light-sensitive detector to detect cells after you've put a fluorescent dye inside the cells. This is kind of cool because the fluorescent dye, you may choose a fluorescent dye that only is taken up by live cells, so you could actually only count the number of live cells. More direct methods to measure microbial numbers. So here, got your flask, got your sample with the microbe in it, and you put it in this unit. Here's the membrane filter. So this, you'll pour the sample, there'll be a vacuum pump, the sample will be drawn through this port, and then the membrane will catch all the cells, and then you can take the membrane and flip it upside down, or you can put it in right side up. It depends on how you want to do it, but either way, the microbes will now grow because this culture medium in the agar, this is what your microbe of interest likes to eat. And then you can count the number of colonies. And you can assume that each colony started with 1 cell. You can use this method when there's a low population density of microbes. So in your original sample up here, the population was really low, like fecal bacteria in a stream or lake. So yeah, hopefully if you're swimming in a stream or a lake, there's not a high count of fecal bacteria. The sample goes over a membrane. We talked about that. Then you incubate it. And then you count the number of colonies. So here's 1, 2, 3, 4. You can count them. That equals the number of colony-forming units in an original large sample. And this is the same thing we were talking about, but this is a light micrograph of the filter. So you can see these little pores here, and then you can see these, it looks like probably is a diplobacillus, because look, it's got the rod shape and you got 2 of them attached. So that's a light micrograph of your bacteria. And then here's just a photograph of what it looks like when you plate those bacteria. This is kind of cool. We've done this before in the lab to use the most probable number to see how many microbes you have. So you take your sample here in this culture tube. It's undiluted, so that means it's in the original, that's the original sample, undiluted. And it doesn't show that you started with 9 mils of media, but you did. And you take 1 mil out of your undiluted sample, put it in here, vortex this, so mix it up, then take a mil and put it in your third culture tube. And then you have 5 more, 1, 2, 3, 4, 5. So 5 culture tubes per dilution. Then you take 1 mil out of your original culture tube and put it in each one of these 5 culture tubes. 1 mil. And these culture tubes have phenol red, that's a pH color indicated. So if this changes from red to yellow, that means the pH has gone down, so the acidity went up. And then you do the same thing for your 1 to 10 dilution, and you do the exact same thing for your 1 to 100 dilutions. And then you look at your results. So from the undiluted sample, you had 4 tubes testing positive, so that were acidic, and 1 was red. In your 1 to 10 dilution, you have 2 tubes positive, and in your 1 to 100 dilution, you have 1 tube positive. Then you take those numbers, 4, 2, 1. So 4, 2, 1, and you use it to look at a chart. Okay. Compare the numbers to a standard chart. So remember, the number is 4, 2, 1. So you come down here, the most probable number table. You can get this. It's in your book, but you can find it anywhere, probably online. And you see here's your series, 4, 2, 1. And then it'll tell you the most probable number of bacteria per 100 mil. So this tells you that you have 26 bacteria per 100 mil of sample or media. So that's kind of fun. So most probable number is just a statistical estimation of the number of microbial cells that you have. It's useful for counting microbes that don't grow very well on solid media, so you can't grow them on those agar plates. If you're using samples of wastewater or drinking water, and there's too few organisms to do a viable count, this is a good way to count them. Okay, this is funny. Microbial social network. Yeah, aren't they cute? They look so friendly, all those little microbes. And they have their social networks as well. They rarely live alone. Microbes rarely live alone. In nature, they form associations. If you're a microbe, you're living in an association. It could be that you have an antagonistic relationship with another microbe, so you harm another microbe. You could have a synergistic relationship with the microbes. Each microbe in a synergistic relationship receives a benefit that exceeds those that would result if they lived apart. So if me and my fellow microbe lived apart, we wouldn't get as many benefits as we would living together. So for an example, HIV and Cryptosporidium, they have a synergistic relationship to amplify their benefits, but just their benefits are amplified, but your benefits are not, because you're hosting both of them. And so that their beautiful synergistic relationship just increases the probability that you die. And so that's an antagonistic relationship, a synergistic relationship, a symbiotic relationship. You live in close contact with each other. You become interdependent, and you can't survive outside the relationship. It's probably not healthy. Microbial associations include biofilms. I've talked a little bit about those. Here's biofilm. This looks like it's a scanning electron micrograph of biofilm on a catheter. Here's Streptococcus mutans biofilm on teeth. So biofilms are really complex communities of a whole bunch of different types of microbes, and they adhere or they stick to an extracellular matrix because they have glycocalyces, they have fimbriae, and they have proteins that allow them to stick to the surface of interest and stick to each other. There's a whole bunch of different types of organisms in biofilm communities. We saw the photograph of dental plaque. Rocks in streams. Have you ever stepped on a rock? It's kind of slippery. It's probably a biofilm. There's biofilms on your shower curtains if you don't clean them. Implanted medical devices. That's not good. There's biofilms in your digestive system. Up to 70% of all infectious diseases are caused by biofilms. So it's probably a good idea to study biofilms. Strep throat is a biofilm. Neisseria gonorrhea, that's a biofilm. And that's the end.